You are walking through a forest. You have been walking since sunrise. Your stomach is hollow. Your energy is fading. Then you see it. At the base of an oak tree, a mushroom, cream cap, pale gills, a faint almond smell. Everything about it says food. Your hand reaches for it. And then the question hits you, the question that has killed thousands before you. Is this safe? No field guide, no writing system, no expert to ask, just you and a mushroom, and a choice where the wrong answer means dying in agony. Your liver shuts down over 3 days. Your skin turns yellow. You bleed internally. You die conscious. That is what a death cap mushroom does to you, and it looks exactly like several edible species. Evolution made it look edible because being eaten helps it reproduce. You are the target of its strategy, but here's the thing: you are alive, which means someone in your deep ancestry got this right. Not just once, thousands of times, across thousands of times, across thousands of generations. How? This is the paradox at the heart of human food knowledge. Without taste, without smell, they look like food, they taste like food, and then they destroy your organs while you sleep. How did any ancient human survive mushroom foraging? How did our ancestors build a knowledge system about fungi? Without microscopes, without chemical analysis, without writing. The answer is more sophisticated than simple trial and error, and it reveals something profound about how the human mind works when survival depends on getting the answer right. The standard story: ancient humans ate random mushrooms. Those who ate bad ones died. Survivors learned trial and error. Painful, costly, but effective. That story is not wrong. But it is radically incomplete, because what archaeologists and ethnomycologists have uncovered over the past 50 years reveals a knowledge system built with genuine scientific rigor. They observed animals systematically. They developed taste testing protocols that minimized risk. They encoded identification data into language itself. They built ecological calendars tied to observable events. They developed processing techniques that transformed deadly toxins into safe food. They preserved knowledge across hundreds of generations through oral traditions so precise that modern mycologists buy that it's highest to diff with the highest possible stakes. Let us look at how they did it. Start with what makes mushrooms uniquely dangerous, because understanding the problem reveals the sophistication of the solution. When an ancient human encountered an unknown plant, they had multiple testing options. Smell it, touch it to your skin, crush a leaf, place a piece on your tongue. Each gives information within seconds. Many toxic plants produce immediate sensory warnings. Bitter tastes, burning sensations, evolution way of telling you to stop. Mushrooms do not cooperate. The deadliest mushroom toxins, amatoxins found in death caps, Amanita phalloides, are completely tasteless and odorless. They cause no immediate symptoms. You eat a death cap and feel fine for 6 to 12 hours. The mushroom tastes pleasant. While you sleep, amatoxins reach your liver and bind to RNA polymerase, essential for protein synthesis. Liver cells die by the millions. By the time you wake, the damage is catastrophic. The only effective treatment is liver transplantation without a hospital. Mortality approaches 100%. This is a food that tastes good and kills you 3 days later. A 2019 Katharina Schenkjäger at the Medical University of Vienna, Clinical Toxicology, analyzed over 5,000 confirmed amatoxin poisoning cases across Europe and found that without intervention, the mortality exceeded 95%. A single death cap contains enough amatoxin to kill an adult. There is no safe test dose, no sensory warning, no second chance. So how did ancient humans solve this problem without dying repeatedly? The first line of defense was animal observation, far more sophisticated than simply watching what animals ate. Giorgio Samarini, the Italian ethnomycologist at the Museo Civico di Roverti, documented how traditional foraging cultures learned about mushrooms by watching animals. His 2001 book Animals and Psychedelics reveals a pattern across continents. In Central America, indigenous foragers identified psilocybe mushrooms by observing goats that ate them and stood motionless for hours. In Papua New Guinea, foragers found edible boletus by watching wild pigs dig them up with enthusiasm. In Siberia, the Koryak observed reindeer seeking Amanita muscaria during specific seasons. But a crucial detail: ancient foragers did not assume that what was safe for one animal was safe for humans. They cross-referenced across multiple species. If a deer, a bird, and a rodent all ate the same mushroom without ill effects, that was strong evidence. If only one species ate it, that was a warning sign. This is multi-species triangulation. Any modern toxicologist would recognize it. David Aurora, author of Mushrooms, Demystified, noted that the most reliable traditional foragers in Mexico and Eastern Europe consistently used animal behavior as secondary confirmation, never as primary identification. They watched animals to generate hypotheses, then tested those hypotheses through their own protocols. The second layer was systematic taste testing, conducted with a graduated protocol that minimized risk. This protocol was so consistent across cultures that ethnomycologists reconstructed it as a standard method. Step 1. The mushroom is touched to the inner wrist or lip and observed for hours. Any tingling or numbness signals danger. Step 2. If no reaction, a grain-sized piece is held in the mouth for several minutes, then spat out. The mouth is observed for another hour. Step 3. If still clear, a grain-sized piece is swallowed, followed by a full day of observation without eating other new foods. Step 4. Only after all tests pass is the mushroom provisionally safe, and even then it is cooked before larger consumption. This protocol appears in modified forms from the Italian Alps to Southeast Asia. Andrea Pironi, at the University of Gastronomic Sciences, documented this protocol in rural European communities, preserved orally for over 4 centuries. His 2006 study in the Journal of Ethnobiology and Ethnomedicine found families using the same testing sequence as their great-great-grandparents, with modifications only in the specific mushrooms, never in the method. This protocol required 3 things not guaranteed in prehistoric life: people willing to put lethal substances in their mouths, a community willing to care for potential poisoning victims, and a system for transmitting results across generations before anyone forgot what happened. The third and most elegant layer was encoding mushroom identification into language itself. Traditional cultures compressed generations of observation into mushroom names that function as miniature identification manuals. In the Zapotec language of Oaxaca, the name for Boletus pinophilis translates to mushroom that grows where the pine resin falls. The name encodes habitat, host tree, and season in a single compound word. In Japanese, matsutake means pine mushroom, encoding the specific host tree. In Russian, the name for Boletus edulis is belegi, grib white mushroom, distinguishing it from the staining, look-alike Boletus luridiformis. These are compressed identification protocols. Terence McKenna argued in Food of the Gods that the precision of traditional mushroom nomenclature across cultures suggests that language itself may have been driven by the need to transmit survival information. The names are memorable, descriptive, and actionable. They tell you where to look, when to look, and how to distinguish safe from dangerous. A 2007 study by David Pegler at the Royal Botanic Gardens, Kew, cataloged over 800 traditional names across 40 languages. Over 70% encoded at least one piece of actionable identification information: habitat, season, color change, or look-alike species to avoid. This is a knowledge transmission system built into the fabric of language, accessible to anyone who speaks it, preserved as long as the language survives. Beyond language, ancient humans built ecological calendars that tied mushroom seasons to observable natural events. In indigenous North American cultures, morel season was marked by trillium. Blooming chanterelle season was tied to the first summer thunderstorms. In Eastern Europe, specific butterfly species signaled the start of porcini season. These markers served the same function as a modern foraging app, requiring no technology or written records. Dr. Harriet Kuhnlein at McGill University documented this knowledge system among Canadian Indigenous peoples in her 19091 study traditional plant foods of Canadian Indigenous peoples. She recorded over 30 distinct ecological markers used by different First Nations to time their mushroom foraging. Different communities in the same region used different markers for the same species, suggesting independent development through careful local observation. Some markers were remarkably precise. The Tsimshian people of coastal British Columbia timed their chanterelle harvest to the exact week when salmonberry bushes produced their first ripe fruit, not flowering, not leaf emergence. First ripe fruit. This precision requires sustained observation, tracking events that are ecologically linked. Chanterelle fruit when soil temperatures and moisture reach specific levels. Salmonberries ripen when temperature and moisture reach the same levels. The indigenous foragers who made this connection were doing ecological science. They just did not call it that. The most dramatic, Archaeological evidence of ancient mushroom knowledge comes from a man who died over 5,000 years ago. In 1991, hikers in the Alps discovered Oetzi, the Iceman, preserved in a glacier for 5 millennia. Among his belongings were 2 types of polypore mushrooms. On leather thongs, one species, Fomentarius, was used as tinder for starting fires. But the second species told a different story. Oetzi carried Piptoporus betulinus, the birch polypore, which modern analysis confirms contains anti-inflammatory, antibiotic, and antiparasitic compounds. Oetzi also carried evidence of whipworm eggs in his intestines. Piptoporus betulinus contains agaric acid, active against intestinal parasites. 5,000 years ago, someone identified a specific fungus on a specific tree, understood its medicinal properties, harvested it, and carried it to treat an infection that modern microscopy could only confirm millennia. Later, Patrick Spindler of the University of Innsbruck, who led the analysis, noted that this provides compelling evidence of targeted medicinal use in the Copper Age. This is not random mushroom picking. This is a person carrying medicine. The knowledge required to make that identification, to know which tree to search, which fungus to harvest, and how to prepare it, represents thousands of years of accumulated empirical observation passed down through oral tradition. Mycologists describe a spectrum of cultural attitudes called mycophilia versus mycophobia. In mycophilic cultures with positive attitudes toward wild mushrooms include East Asia, Eastern Europe, Russia, and the Mediterranean. Foraging is a normal family activity. Children learn identification from a young age, and knowledge is actively maintained. Mycophic cultures include the English-speaking world and Northern Europe, where wild mushrooms are viewed with suspicion and foraging is a niche hobby. David Aurora proposed in Mushrooms Demystified that this divide is driven by the local abundance of signature mushrooms. Species so distinctive they are nearly impossible to confuse with lookalikes. Matsutake in Japan, with its distinctive spicy aroma, porcini in Italy, with its net-like stem, chanterelle in Eastern Europe, with their trumpet shape and apricot smell. Where signature mushrooms exist, the reward for learning identification is high and risk is low. Where they do not exist, the risk-reward calculation shifts dramatically. Quote, a 204 FAO report by Eric Boa surveyed mushroom knowledge across 120 countries. It found a direct correlation between signature mushrooms and the depth of traditional mycological knowledge. In cultures with signature mushrooms, over 90% of adults could correctly identify at least 5 edible species. In cultures without them, that number dropped below 20%. One of the most impressive achievements of traditional mushroom knowledge is processing techniques that transform deadly species into safe food. The false morel, Gyromitra esculenta, contains gyromitrin, which breaks down into monomethylhydrazine, a compound used as rocket fuel and a potent neurotoxin. Yet Gyromitra esculenta has been eaten in Eastern Europe and Scandinavia for centuries. The traditional method involves a precise multi-step process. First, the mushrooms are boiled in a large volume of water. The water is drained and discarded. The mushrooms are boiled again in fresh water. Then they are thoroughly dried. Then they are cooked again before consumption. Modern analysis confirms this process reduces gyromitrin by over 90%. The Finnish Food Authority still recognizes this method as safe. And false morels remain a prized seasonal food in Finland. What makes this remarkable is not just that someone figured out the method, but that they did so through experimentation. Someone had to try boiling once and test the result, then boiling twice, then boiling and drying. Each iteration took days and carried risk. The knowledge that all steps were necessary had to be preserved accurately. A single mistake could kill. Dr. Juhani Ruotsalainen at the University of Eastern Finland studied this tradition and found that written records of the method extend back to the 100 survived. The knowledge survived for hundreds of years, despite the lethal consequences of error. Ancient mushroom knowledge extended beyond identification and processing to deliberate cultivation. The cultivation of shiitake, Lentinula edodes, was developed in China and Japan over 1,000 years ago. The method requires a specific sequence. Hardwood logs are cut in winter when sap content is lowest. They are moved to a shaded, humid location, Holes are drilled in a specific pattern. The logs are inoculated with shiitake spawn and left for 6 to 12 months, while the mycelium colonizes the wood. Then they are soaked and leaned to trigger fruiting. Each step requires precise knowledge developed through systematic experimentation across generations. The tree species, cutting timing, hole pattern, colonization duration, and soaking time all affect yield and quality. A 2002 study by Dr. Daniel Roys at Pennsylvania State University, Bioresource Technology, analyzed traditional shiitake cultivation and found that the optimal parameters identified by Japanese cultivators over a millennium ago closely match those identified by modern mycology. The traditional method is not folk tradition. It is empirically optimized agriculture developed through centuries of deliberate experimentation. The same pattern appears with other cultivated fungi. Paddy straw mushroom cultivation was developed in Southeast Asia over 2,000 years ago. Using rice straw as substrate, oyster mushroom cultivation emerged in Europe. Using coffee grounds and agricultural waste, a method rediscovered by modern mycologists in the 1970s, the pre-Columbian peoples of Mexico developed knowledge about psychoactive mushrooms, so detailed that Western science took centuries to catch up. Gaston Guzman, the Mexican mycologist who documented traditional mushroom knowledge in Mesoamerica, recorded over 50 psilocybe species used in indigenous ceremonial practices. In his 2008 economic botany study, Guzman noted that traditional Mazatec and Zapotec foragers distinguished between morphologically nearly identical psilocybe species based on subtle differences in habitat, spore color, and bruising reaction that Western mycologists initially missed. The knowledge was encoded in a naming system predating European contact by at least 2,000 years. The Mazatec name for Psilocybe cubensis translates to the mushroom that makes you see the truth. The Zapotec name for Psilocybe carolicens translates to the mushroom that speaks through the ear. These names encode information about each species' subjective effects, allowing users to select specific mushrooms for specific ceremonial purposes. Jochen Gartz at the University of Leipzig confirmed this accuracy through his 195 chemical analysis of traditional Mexican psilocybe species. The alkaloid profiles corresponded closely to the effects described. In traditional names, cultures operating entirely outside Western science had developed a pharmacological classification system that took modern chemists decades to replicate. Spanish colonization suppressed these traditions as pagan practices, but knowledge survived through oral transmission in remote communities, rediscovered by R. Gordon Wasson in the 1950s. The most surprising example of ancient mycological knowledge comes from Siberia, where the Koryak people developed a sophisticated understanding of Amanita muscaria, the iconic red mushroom with white spots. Amanita muscaria contains ibotenic acid and muscimol, producing altered perception and trance-like states. The Koryak discovered that the active compounds are largely excreted, unmetabolized in urine. They developed the practice of drinking the urine of someone who consumed the mushroom to produce a second psychoactive experience, recycling compounds that would otherwise be wasted. This was especially important because Amanita muscaria was scarce during certain seasons. Modern pharmacology confirms that muscimol is excreted, largely unchanged in urine, and the concentration in the urine of someone who ate the mushroom is sufficient to produce effects in a second person. The Koryak did not simply know the urine was active, they knew the timing, dosage, and conditions for effective use. Multiple Siberian cultures, the Koryak, the Chukchi, the Evenki, independently developed this practice and transmitted it orally. Dr. Rain Willislev at the University of Oslo documented these traditions in his 2000 ethnographic study. He noted that the accuracy of traditional knowledge about Amanita muscaria pharmacology was so high that modern researchers initially struggled to believe it could have been developed without chemical analysis. Yet the evidence is clear. These cultures empirically verified the metabolic pathway of a complex psychoactive compound through careful observation, thousands of years before modern biochemistry confirmed their findings. This knowledge system is now being lost at an alarming rate as people move to cities and processed food replaces foraged food. The mushroom knowledge accumulated over tens of thousands of years is disappearing within a single generation of urban migration. The consequences are measurable. In the United States, the American Association of Poison Control Centers reports over 7,000 confirmed mushroom poisoning cases annually. Most involve people who misidentified mushrooms using identification rules from home countries that do not apply to North American species. A Vietnamese immigrant in Oregon identified a deathcap as a species edible in Southeast Asia. A Polish immigrant in Michigan collected a look-alike that does not grow in Poland. The knowledge that kept their ancestors safe is encoded in specific languages, tied to specific ecosystems, and preserved through cultural practices that are not transmitted when people move. Michael Bugg at Evergreen State College, who studied North American mushroom poisonings for over 30 years, documented this pattern in his annual reports. Immigrants from mycophilic cultures are overrepresented in serious poisoning cases because they apply knowledge specific to one ecosystem to a different ecosystem with different species. The knowledge is not wrong, it is context dependent, and when context changes and knowledge does not update, people die. Every language that loses its traditional mushroom vocabulary, every family that stops foraging, represents the end of an unbroken chain stretching back into deep prehistory. Let that sink in. Someone in your deep ancestry volunteered to put a piece of deathcap mushroom in their mouth, so you would not have to. Someone else watched them for 12 hours. Someone else remembered the result. Someone else encoded it into a name. Someone else taught that name to their children. Someone else carried it across a continent. Someone else taught it to your grandparents, and someone taught it to your parents, and now you know it without ever testing it yourself. The cost of that knowledge is measured in lives lost before the system worked. But the system did work. It worked so well that you walk and sell without ever knowing which species killed your ancestors while they were figuring it out. By Robert Gordon Wasson, the Journal of the Botanical Research Institute of Texas. Trace the oral transmission of specific mushroom identifications across 19 generations. In a single Mexican community, the accuracy remained consistent across all 19 generations. No written records, just human memory. Consider what this means. The next time you eat a mushroom, you are participating in a knowledge system that has been operating longer than any written language, any government, any religion on earth. You are the inheritor of a system, built one observation at a time, tested with every meal, and preserved through the most fragile and powerful transmission mechanism ever evolved, the human voice, speaking to the human ear, telling the next person what the last person learned. The knowledge you carry in your head about what is safe to eat was paid for by people whose names you will never know, whose faces you will never see, whose voices you will never hear. But they spoke, and you listened, and that is why you are still here. You eat history.